Hi class. I just wanted to make a quick video to help walk you through the Amoeba Sisters worksheet a little bit. Enzymes can be kind of challenging and tricky to understand, so I thought I'd walk you through the worksheet just so that you would know what I'm looking for. So this worksheet you're answering it either while you're doing your Edpuzzle video with the Amoeba Sisters, or you can use the video. It's the same video here, it just doesn't have the Edpuzzle questions in it. So you do have to do the Edpuzzle version, but whether you do the worksheet during the Edpuzzle or after, that's up to you. But you are going to open the document with Cami. I already have it open. And so here's number 1. It says, in the box below, please illustrate an enzyme and substrate. Label the following keywords in your illustration. You need to label the enzyme, the substrate, and the active site. In the video, I think they use Pac-Man. So use a simple shape like that. You can use your drawing tool. I'm going to make the line a bit thicker. We can make him yellow like Pac-Man, and just kind of, do your shape, and then you have to have a substrate, so I'm going to draw something different here. My substrate is going to have the right shape to fit into the enzyme. So now you want to label, get a little, go away, a little smaller. We're going to label the enzyme here, and then this is our substrate, and the active site is right here. That's kind of how I would like you to do it. So this V-shaped area on the enzyme, that's the active site. Okay, so you can do something like that. So then here, enzymes are typically which type of biomolecule? I'm looking for you to write either the word carbohydrate, protein, nucleic acid, or lipid. Okay, and you can get that from the video, from your notes. And then you're going to describe the effects that enzymes have on substrates. So enzymes are going to work on a substrate. They do 2 different types of reactions. They can either do a digestion reaction where they break them apart, or they can do a synthesis reaction where they create something bigger. But the video goes into really good detail with that as well. So in order to function efficiently, enzymes need to be at an ideal, which is like the perfect, pH and temperature. Different enzymes have different ideal pH and temperature conditions. If the pH or temperature is extreme for a particular enzyme, it can even denature an enzyme, which means make it fall apart, okay, break it, which can prevent it from binding and acting on its substrate. So for the following 2 scenarios, name the variable, temperature or pH, that is affecting the function of the enzyme. So that is what I want you to look at, is the variable. You are writing temperature or pH on your line here and here. So this one says ATP is produced by cellular respiration in your human body cells. We learned about ATP. That's a type of nucleic acid, remember, that has the 3 phosphates on it. There are a variety of enzymes that work to produce ATP, but one of those enzymes is called phosphofructokinase 1. It's a big word. This enzyme is sensitive to blood acidity. That's a cue or a clue. Blood can become more acidic if a patient is in respiratory distress. So what do you think they're talking about affecting this enzyme? Is it temperature or is it pH? I think it's pretty obvious, but I'll let you write that one down. Our next picture, these people at a grocery store, it says, so you like liver, huh? And I think it'll make more sense when we read the story here. A popular lab that can be performed by students is to test the reaction rate of Catalase enzyme when it acts on the substrate hydrogen peroxide. Catalase has the ability to break down hydrogen peroxide. You might even have hydrogen peroxide at home in your medicine cabinet. Sometimes you use it to clean out a wound or something. So I'm reading here, Catalase can be found in beef liver from the grocery store. So this is an enzyme that exists in beef liver, and like in AP classes, they actually grind it up in a blender and use that in their reaction. However, if the beef liver is boiled first, the catalase will not be able to break down hydrogen peroxide. Well, boiled is going to be important for us to answer this question. So they did something to the liver, they boiled it. Is that relating to temperature or pH? Which variable is affecting the enzyme function? I'll leave that up to you to figure that one out. On the next page, it says, please fill in the chart for every real-life scenario listed below. Some boxes have been filled in for you. So up at the top, we want to identify the name of the enzyme. And then we want to identify the substrate. The substrate is the thing that's getting either made or broken apart. Illustrate the scenario. Label the enzyme and the substrate in the illustration. Illustrate means draw a picture. And then describe the relationship between the substrate and the enzyme in the scenario. And the relationship I'm looking for is the enzyme making something? Is it a digestion reaction or a synthesis reaction? So the first one says lactase. Remember, ASE is indicative of an enzyme. Lactase is an enzyme that breaks down a sugar found in dairy products known as lactose. So anytime you see OSE, they're talking about a sugar. ASE, we're talking about an enzyme. Luckily, our enzymes many times are named for what they work on or break down or build. So it says some people are lactose intolerant, and this can be due to not having enough lactase production. So people who are lactose intolerant may not feel well after eating foods containing lactose. So the enzyme, remember we're talking if it has ASE in it, that's what we're looking for as far as the enzyme name. So we do have lactase right here. They already identified the substrate for you, lactose. And then you're going to draw a picture of what's happening. And it's talking about breaking it down. Okay? So you have to decide, is that digestion or is that a synthesis reaction? So we're trying to decide here, digestion or synthesis. What kind of reaction are we looking at there? Okay. And then the next one, an enzyme called glucocerebrosidase breaks down a glycolipid in the body known as glucocerebroside. However, in genetic disease known as Gaucher's disease, the body does not produce enough glucocerebrosidase. That's a hard word. Therefore, glucocerebroside can build up in the body and cause serious side effects, such as anemia and swelling of the liver and the spleen. Okay, these words are very big and very hard, but I gave you a clue, okay? If it ends with ASE, that's your enzyme. So you have to look through, there's some big words in here. Right here, ASE. So there's your enzyme. And it breaks down, so there's our other clue, it breaks down this one here, okay, glucocerebroside. So you don't have to know, what these words are. You just have to be able to identify that word ending. Oh, that's the enzyme, okay? And again, you're going to draw the picture in the third column and tell me, is it a digestion reaction or is it a building, like synthesis reaction? And then the last one, pancreatitis, so your pancreas sits right under your stomach, is an inflammation of the pancreas which can damage pancreatic tissue. The pancreas produces digestive enzymes such as amylase and lipase. These enzymes assist in breaking down certain food biomolecules. In this disorder, enzyme production from pancreatic tissue may be stopped. So, our enzyme, they are telling us that pancreas produces digestive enzymes such as amylase and lipase, and they're going to break down certain food biomolecules. I'll let you try and guess. What do you think? Amylase and lipase will will be, like breaking down. So over here they did give you its relationship. So it says, since the pancreatic tissue can be damaged in this disorder, the production of the enzymes in the tissue, amylase and lipase, giving you the answer there, may be disrupted as well. This would affect the ability to break down certain types of food biomolecules, the substrate. Now they did give a very long explanation. I want you to focus on, is it a digestion or synthesis? And you can explain the enzyme lactase breaks down and digests lactose, which is the substrate in your stomach. That would be a good way to answer that. So you don't have to go fancy like they do. But that's it for this worksheet. So I thought I'd just kind of help guide you through, because it can be a little tricky to understand what we're looking for in these answers. Again, if you have any more troubles, let me know. Just email me or send me a reminder, and we'll see you soon.